I'd like to begin with, again, thanking Arijit for putting together such a fantastic meeting. It's great to see many faces who have actually inspired me to go in this area when I was wandering as an undergrad trying to find a home, what exactly to work on. So it's really inspiring to have many of them here and learn about the latest things that they have been doing. And also I'd like to thank the student team for putting together the entire conference. So what I know I'm against multiple odds. Of course, an excellent sequence of talks that has gone, Power Play 3, which is about to start, and of course, travel of various people. So I'll try to say something useful in the next 20 minutes or so. And the story that I'm going to tell today actually was a presumed coding mistake. I thought about it, Kishore, my student, is here. We thought about it for a long period of time that this has to be a coding mistake. This cannot be a result. But eventually we did figure out it was not a coding mistake, and there was something fundamental that we had to say about the structures of networks that we have been looking at. So our group focuses on trying to understand cancer from a systems biology approach, thinking about how cancer cells make decisions and these kinds of cartoons. This is, of course, from the most cited paper in the field of cancer biology, Hallmarks of Cancer. And I'll come back to this at the end of the talk as well. It's a very clearly very pictorial, schematic representation, but it doesn't have any quantitative details, which often goes into figuring out a kind of models you'll see. So what we try to think about is how do regulatory networks, they converge to some discrete outputs because they have all these different inputs coming in, which of course vary as a function of space and time. And also thinking about why are networks the way they are, why are molecules connected in this specific geometry as compared to various other topologies that could have been possible. Why did evolution select for these kinds of structures? So the system that we have been working on for quite some time is in the context of cancer metastasis. So if you think about the most common solid tumors, breast cancer, liver cancer, lung cancer, etc., these are all epithelial organs. So cells do not have the inherent property to start moving and start invading. So they undergo this transition where they take on some of these properties which are typical properties of mesenchymal cells, and this transition is called epithelial to mesenchymal transition, which is often helpful in enabling cancer cells to leave their primary tumor. And once they have reached the other organ, they no longer need to invade, they no longer need to migrate, they need to settle down and grow a secondary tumor there. And that is why when they undergo presumably this exact opposite transition of mesenchymal to epithelial transition. So for a very long period of time, EMT or MET had been thought of as a binary process, and that was the first question we asked. This is when I was doing my PhD, that is EMT really a binary process? And at that point of time, we had information about what transcription factors, what microRNAs, how they regulate EMT and MET. So these two transcription factors drive EMT, and these two transcription factor drives MET. And of course, there are various other gamut of molecules also coming into here. And what you see here is interconnections between them in terms of activation, inhibition, transcriptional or post-transcriptional in nature. So we made a simple mathematical model for this network, and what we predicted was that EMT is not a binary process. You could have this hybrid state somewhere in between, between completely epithelial and completely mesenchymal, and this hybrid state could have functional relevance as well, that it had the capability to migrate while being still adhered to some of its neighbors, giving rise to a collective or a cluster type migration as compared to a individual type migration. So there were multiple predictions that we made at that point of time. A, EMT is not binary and cells can exist stably in this state. That even if you start from an isogenic population with the cells with identical genetic background, you could still have multiple phenotypes. And of course, as it happens in any multi-stable system, cells can switch back and forth given enough noise. So we started looking together with experimental collaborators to validate these. So these are lung cancer cells co-stained for E-cadherin and vimentin in green and red respectively, which are canonical epithelial and mesenchymal markers. So what you see here is that over multiple passages in the lab, individual cells continue to co-express epithelial and mesenchymal markers. You can quantify this in much more detail. You can look at whichever cell line of interest. This is breast cancer, prostate cancer, lung cancer, colorectal cancer, and you do see that that despite identical genetic background, there are cells in all of these 3 different quadrants. And the other set of colleagues did these experiments where they segregated these 3 quadrants of subpopulations and cultured them independently in the lab. And what you see is that each of them was able to switch to everyone else, therefore giving rise to phenotypic heterogeneity that emerges as a consequence of this. We are not the only ones saying that EMT is non-binary. There have been multiple mathematical models that have come up in past 10 years. This is an extended ODE-based model. People have been doing Boolean models as well, projecting them on 2 dimensions of epithelial and mesenchymal. And clearly there are these extreme states, but there are many of these hybrid states, depending on what network you're looking at, what is the size of that, and so on. And this is similar other case where you see that there are epithelial and mesenchymal states on this principal component plot, but you see a whole multitude of these hybrid states as well. So in the past 10 years or so, there has been extensive modeling data, experimental data, clinical data, all saying that EMT is not binary. You can have cells in this entire continuum. Multiple states exist. They exist in vitro, in vivo. This is data from circulating tumor cells. And not only they exist, but also they play a crucial role in metastasis. There have been studies saying that if you can eliminate particularly those hybrid cells, you can bring down metastasis dramatically. And this was a recent study showing where they were actually quantitatively comparing what is the metastatic ability along this entire spectrum. And you see this non-monotonic response that it increases in between in these hybrid states and then again settles down. So in other words, these hybrid states actually have many of these properties which make them perhaps the fittest for the process of metastasis. I'm summarizing a lot of literature here, experimental as well as clinical, which has come out in the past decade, showing that these hybrid cells can evade the immune system, form tumors, resist chemotherapy, associate with worse survival. And interestingly, what caught our attention was a more quantitative measure saying that these hybrid cells are more plastic, or they're easier to switch to another state as compared to when you start from completely epithelial or completely mesenchymal. And this is the work which Kishore, my PhD student here, who was here in the audience, started looking at, that if people are consistently reporting that these hybrid cells are easier to switch from, why is this observation being so consistently there? Is this high plasticity behavior an emergent feature of the networks that underlie? And if yes, then can we identify those network features without doing any numerical simulations of the networks that we are going to look at? So the networks, of course, in the past 10 years have grown much larger. We started with the 4-node network, and you can clearly see the networks now which are available in the literature. None of them was put together by us, just to avoid any bias in terms of which system we are looking at. These were put together by 4 different groups. And all we have done here is just labeled epithelial and mesenchymal nodes based on what's known in the literature into red and blue respectively, and edges show green is activation and red is inhibition. So we did Boolean models for this because these are large dimensional systems. Doing ODE becomes computationally very expensive. And in the Boolean models, again, we applied a simple majority rule saying that for a given node, you just count the number of activations and inhibitions that come onto it. If number of activations is more than inhibitions, you turn the node on. If number of inhibitions is more than activations, you turn the node off. And if they're equal, you don't update the node, you just let it stay wherever it is. And every node can take only 2 values, on or off. So it's a very simple model in that sense. We are compromising on a lot of biological detail here, which clearly we could have gone into and made the model more accurate. So what we see when we simulate this system, we notice that epithelial and mesenchymal states tend to be the most dominant states, but you get a whole bunch of these hybrid states where some epithelial nodes are on and simultaneously some mesenchymal nodes are on. In contrast to the epithelial and mesenchymal, where you can see that in the mesenchymal state, all mesenchymal nodes are off, all epithelial nodes are off and all mesenchymal nodes are on, and vice versa is what you see for epithelial. So epithelial and mesenchymal are clearly very antagonistic patterns, but hybrid, there is no one hybrid state. There are multiple of these states. So we wanted to quantify this in more detail. This is one of the metrics we looked at, coherence, where what we have done is, and we looked at this across all different networks. We start with a given steady state and perturb one of the nodes, either on to off or off to on, and then see whether the system comes back or it goes away. And then you can quantify that on a scale of 0 to 1 depending on how many times it comes back. So you can clearly see across networks there was a clear bimodal distribution, that there were some states which had low coherence and then there were other states which had relatively high coherence. You could also define frustration. This is again taking directly from physics, that frustration is basically the number of times that a state does not obey a topology. So if A activates B and A and B are both on, it is not a frustrated state because both of them are on and that obeys the rule between them. But if one is on and the other is off, and A is supposed to activate B, then this disobeys the rule, so therefore we call this a frustrated state. So again, you can quantify this on a scale of 0 to 1. So then when we projected coherence and frustration together, across networks we saw a very clear trend. Both of them were bimodal in nature and they were strongly negatively correlated, which makes a lot of sense because if you are more frustrated, then if you are given a perturbation, it's more likely that you would actually go away rather than coming back to the same point. Putting things together, coherence and frustration I've already introduced. All we did here was overlay steady state frequency on top of it. And what we see is a very clear pattern, that the epithelial and mesenchymal states are the ones which are highly coherent, less frustrated, and the hybrid states are the ones which have very high frustration and less coherence, and they are the ones which have the least frequency. So this can possibly explain why all these experimental observations, that if you're in a hybrid state, you're already very frustrated, all you need is a little nudge to leave that state. But if you're in an epithelial and mesenchymal state, that nudge needed would be much higher. But again, all we have done here is said things in a more quantitative way. We have still not explained why are hybrid states more frustrated. Is there something inherent in the structure of the networks which actually allows for this kind of feature? So to look at that, what we did is we took one of these networks, and we have done for other networks as well, of course, and we converted this network into an adjacency matrix where each row is the source, each column is the target. So in this 22 cross 22 matrix, 82 elements are filled because these are direct edges, either +1 or -1, and the remaining ones are all empty. But as we very well know, both in biological networks as well as in life, you can be influenced by another individual even if you're not in direct contact with them. This can happen through WhatsApp messages, this can happen through multiple intermediaries that you have. So all we have done here is we just multiplied this matrix to itself multiple times, and we see a very clear structure. That every epithelial node activates every epithelial node, every mesenchymal node activates every mesenchymal node, every epithelial node inhibits every mesenchymal node, and every mesenchymal node inhibits every epithelial node. So this is a very clear structure that sort of comes up here, which is what we call the phenomena of teams of players. So there's no one EMT inducer or there's no one MET inducer, but there's this whole bunch of players supporting each other on one side, supporting each other on the other side, and then they're playing a tug of war between them. So I wanted to ask that whether the formation of these teams, is it unique to the networks that we are looking at, or this could also be seen in any random network of given size and given density. So to look at that, what we did is we generated negative controls, which is that you have the same network size, same number of edges, but the adjacency matrix, you flip the values. You generate many such hypothetical non-biological random networks, and then you quantify the team strength on a scale of 0 to 1. The wild type network has a team strength around 0.7, and these are 1,000 random networks, and you can see that the wild type network lies somewhere around 90 to 95 percentile. You can change the network. The wild type network on this is lower, it's 0.25, but as compared to its randomized counterparts, that value is still around 95 percentile or higher, saying that the presence of teams is largely unique to the networks that are actually existing in nature. Then the next question we asked was, sure, teams exist for epithelial and mesenchymal, but is this the reason why hybrid states do not have as high stability? So to quantify this further, what we have done is we started with an epithelial, mesenchymal, or a hybrid state and asked the question, on average, how many nodes would you need to flip from on to off or off to on so that you leave the state that you started from? So what is the nudge that you would need to give to that particular state so that it loses its identity and it goes to a different state? When you start from epithelial or mesenchymal, that number is around 40%. When you start from hybrid, that number is 5%. So there is an order of magnitude difference between the stability of these states, which can actually be quantified here. And again, you can look at a random network which does not have the team structure, and that behaves very differently than this wild type network that follows this clear sigmoidal like pattern here. So again, this is actually happening due to the presence of teams. So what we are saying is that because there are strong teams in certain scenarios, that is what restricts how many hybrid states you can have. But when you begin to have teams breaking down, then is when you can have many, many more hybrid states. And the reason that there are no teams for hybrid states is what makes them the most plastic, is what makes them the fittest for metastasis. So next question comes is, are teams seen in other examples of plasticity, or is this just an example that we saw in EMT? So this is lung cancer, and this is where we thought the coding error was, but it was not actually, and this is how the entire idea started before we looked at EMT in more detail. So what we have done here is, in this 33-dimensional system, we solved the coupled ODEs over multiple ranges of parameter values and initial conditions, and then look at all these pairwise correlations. So you can clearly see that there are this one team of 22 players and this another team of 11 players which are mutually antagonistic, but within them they are strongly positively correlated to each other. You can look at the actual data from cell lines of this particular cancer. You see very similar pattern being reported when you look at these 33 genes. You can change the cancer, you can change the genes. This is now melanoma. Again, you see that there are these team-like structures. So one team supporting a more proliferative behavior, another team supporting a more invasive behavior, and again, a strong mutual antagonism between them. So the next question that comes is, sure, even if teams exist, what is their possible function? So one idea is sort of straightforward, that they provide robustness. So if you start from a given network and you do in silico CRISPR, which means that you delete an edge or you change the sign of an edge, the network structure or the network phenotypic behavior should not change as much. And that's what we see. Only 12 out of 357 edges impact the distribution. The network is robust to 345 perturbations. That's around 97% of the cases. The next analysis was, does teams also constrain the number of steady states that you get in a structure? So when we look at the random networks of this counterparts for this network and plot the number of steady states, the distribution here shows that the median value is somewhere around 1,000 to 10,000 states, but in wild type you get only 4 states. So just because of the presence of teams, you can reduce the dimensionality all the way from 10 per 4, 10 per 5 to the case of 4 dimensions here. So I wanted to look at this more carefully, and this work was led by 3 students, Kishore, Pradyumna, and Ashna, asking that can teams also contribute to dimensionality reduction when looking at transcriptomic data. So we simulated the solutions of all these steady-state solutions of all this network and projected them on principal component axis. PC1, principal component 1, explains 95% of the data, which was very, very surprising to us. You can look at the random networks, and that PC1 variance value lies somewhere around 0.2 to 0.4. And you also see that the higher the team strength in all these random networks, the higher the principal component variance. So as long as you can control the team strength, you can actually control how much data is being explained by PC1. Now, what is the constitution of PC1? Every mesenchymal node contributes positively, every epithelial node contributes negatively. Very clear segregation that you see. You can look at a random network and that structure is clearly disrupted. So both the transcriptomic data, which I'll just show in a minute, and EMT networks seem to be explained largely just by the presence of principal component 1. We looked at small cell lung cancer that I showed you earlier. We looked at 2 developmental biology examples as well for which the networks are available. And when we look at generate their influence matrices, you see the presence of teams. You also see PC1 being high. You see that the contribution of PC1 is coming from team 1 in the positive, team 2 in the negative. And consistently, you see that the wild type networks in each of them have the highest team and also the highest PC1 variance. So all the patterns that you saw in EMT networks were consistently seen in multiple other networks. None of this was generated by us. The networks were generated by us. Let's look at transcriptomic data. So this is from small cell lung cancer. These are EMT. So we took the real data from multiple cell lines, and again you see principal component 1 is reasonably high in both of them, and all the genes that you earlier talked about, they continue to form this strongly antagonistic pattern, which is basically 90-degree rotation of the waterfall plot that Rukmani was talking about consistently. So when we saw this in many different contexts, we wanted to ask, can we generalize this even further? So here what we have done is generated artificial networks. So let's say there are two teams. Everyone can activate within each team, and everyone across teams can inhibit, and you can place the edges as you want for different density. And the question we were asking is, how many edges on average would you need in a network to give you a high team strength? And that seems to follow a clear sigmoidal pattern, that as long as you have 20% of the edges, you already have 50% of the team strength. So, and once you reach around 30 to 35%, your team strength is more or less maximal to what you were here. So clearly with 1/3 of the edges being present, you can have the maximum team strength that the network would allow for. So why would networks have higher edges than that or higher density than that is a question that still needs to be looked at. Similarly, about principal component 1 variance, again it's a sigmoidal where the cutoff, you can reach principal component as the highest value around 25 to 30%. And you can look at principal component 1 and team strength, and you see a very similar behavior. So all of this is suggesting is that the presence of teams which was clearly, I showed you both in regulatory networks as well as transcriptomic data in multiple different contexts. Even in EMT we looked at multiple different networks. It seems to be a more generic feature of cases when a decision has to be made and when a cell has to decide which path to go through when both of those paths are mutually exclusive in nature. The other question we started thinking about is, can the presence of teams also contribute to metastasis? Now, the journey of a cell undergoing metastasis is highly challenging, and the success rate of this process is less than 1 in 10,000 cells. The reason for that is because the environment around a cell is constantly changing, and these cells need to undergo many transitions. They need to navigate multiple bottlenecks, some of which are listed here, so that they can eventually pass through those bottlenecks and survive and eventually cause patient death. So the idea was that maybe there are teams comprising of EMT and therapy resistance. So when cells begin to move, they already have turned on drug resistance, and therefore they are fitter during the process that they can tackle these changes. So one example we looked at here was indeed of this exact scenario. This is a drug which is the first targeted therapy that is given to a large percentage of breast cancer patients, and this is called tamoxifen, and there is literature that certain patients do not respond to this drug at all. There's some evidence that EMT, the process that I talked to you earlier about, contributes to this resistance of drug, but we wanted to ask a more causal connection. Does a causal connection exist between these, or are these just two independent processes? So again, we put together a small network which has players of EMT, which has players of tamoxifen resistance, solved it over multiple initial conditions, get some steady states. Largely speaking, what we found was that the higher the EMT, the higher the resistance score. But again, this is only an association, so we next looked at causative connections, asking what happens when you drive EMT. So our model predicted that the resistant population should go up, and then we looked at transcriptomic data when EMT was induced in multiple different forms, in multiple different cell lines, and you can clearly see that EMT goes up and estrogen response, which is the molecule that this drug targets, goes down. So because this signaling pathway has gone down, the drug will not be able to impact the patients. Vice versa, when you induce resistance by either depleting cells of this molecule or generating sublines of this particular, by exposing it to increasing concentration of the drug, you see EMT goes up. So process A drives process B, and process B is driving process A. So through either of these scenarios, this is the formation of teams that is happening, is that they are enabling both the process to go simultaneously. So clearly, patients who are receiving this drug are actually having a major side impact. What we do not have are numbers here in terms of how many cells get killed and how many cells become more metastatic as a consequence of the treatment that is given to them in the clinic at this point. So what we are thinking at this point is that the idea of teams of nodes can perhaps be one of the scenarios that can underpin how cells, just as in the case of protein structure, we minimize energy, the presence of teams can minimize frustration in gene regulatory networks, and this might be one of the underlying principles, how a cell which has multiple options to in terms of phenotypes to take, ends up taking only about 200 options. So how is that huge dimensionality reduction happening from order of 10 power 12, 10 power 14, all the way to 10 power 2 is something which we currently are working on in more detail. So clearly there are open questions. How do we rank team members based on their contributions? We know, again, both in life and in biological networks, if you have members of a team, they are not necessarily contributing equally. So how do we rank them? And how does team membership scale up when you look at much larger networks? And again, what happens when you have more than 2 teams competing for the same thing? And when you have more than 2 teams, but you have to pick only one winner, then how is that hierarchical model being set up such that some options keep getting eliminated through equivalent of knockout matches is what we are still thinking about more carefully. So I'll just summarize here. That the networks that we looked at driving cell state switching, all of them did show multi-stable dynamics, which can possibly explain why you see phenotypic plasticity and heterogeneity. And the existence of teams was also a common feature in networks as well as transcriptomic data, which could explain this canalization of phenotypes. And we currently think that teams can be a meaningful way to reduce dimensionality in the system rather than just going at it in a blind manner. I'll just end with this cartoon that I started with. So clearly this cartoon is a great starting point, but it does not have all this extra information which we would need to make it predictive quantitative models. So and this can have the lack of this information can actually have a major clinical impact, such as what happened with the clinical trials when anti-angiogenesis drugs were given, which is that you stop the blood vessel growth, and in some patients that actually drove higher metastasis, because when you block blood vessels, cells are becoming more migratory because they don't have nutrients around them. So we need to think about cancer as a complex system, and this is not just coming from me. These are actually the last sentences of the most famous reviews in the field of cancer biology. This is almost 25 years ago now, saying that one day we imagine that cancer biology will have a logical coherence that rivals that of chemistry or physics. And 10 years later, when they wrote Hallmarks of Cancer, the next generation, they say, the same authors say that we continue to foresee cancer research as increasingly logical science in which some of these underlying organizing principles can be potentially identified. So we think that the presence of teams, both in developmental biology and in cancer biology networks, might be one of those underlying organizing principles through which we can classify phenotypes more accurately and also look at transitions in a much more quantitative predictive manner. With that, I would like to thank the fantastic set of people I've had the fortune to work with, who again come across different disciplines and enrich our understanding in multiple ways, and the team of students who also represent different backgrounds and disciplines and work towards this. I've also recently taken over as editor-in-chief of this particular journal, NPJ Systems Biology and Applications, and I'll be very happy to discuss if there are any articles that you would like to consider submitting here. Thank you so much once again for your attention. I'll be happy to take questions.
Questions? Yes. Yeah, about, so very nice talk. So one of the early slides on the teams where you showed the adjacency matrix and then you raised it to some power and so on. Did you bi-cluster the adjacency matrix upfront, or were they clusters like there? And I mean, the ordering of the...
Right. No, we didn't bi-cluster it. We looked at, once we arrived at the influence matrix, then we did hierarchical clustering on the influence matrix. And
if
then,
you can go back to that slide.
Sure.
So what is the order of the rows in this?
Yeah, so once we obtain teams here, then we just use the same order of the rows to show the adjacency matrix.
I think I misunderstood, because I thought you obtained the teams by squaring this matrix or raising it to a high power.
Right. So here, the order is because we already have the information about which nodes are epithelial and which nodes are mesenchymal. Okay. So we arrange them together simultaneously.
And then you raise that to some power, and then you get that matrix?
Yes.
And then from that you reorder this matrix? Or?
No.
So structure is coming from, so no, my question was how the rows, KLF8 and then TCF3, and then, how was that order?
Right, so these
defined?
are all mesenchymal, all epithelial nodes together because we already have that information.
Okay, and in your random networks, they are not necessarily together.
Yeah.
And therefore, if you were to bicluster the random network first, you might get a structure like this, but just taking it as it is, you don't.
Yes, yes.
Okay, thanks. Yeah.
Nice talk. So I'm just wondering, with respect to your question, is more of a remark. So when you say that you have more membership in your teams than what is required for the team strength, is it because you want to maintain the robustness of the teams? You have some substitutes already encoded. So by substitutes, I'm talking about biochemical redundancy in terms of paralogs being present. So have you looked something along these lines?
That's an absolutely fine hypothesis, and that's what we also currently think, that because there's a cost to establish every edge, there has to be some benefit, and then benefit perhaps has to be in the context of robustness. So that analysis is still ongoing, and we are looking at larger networks to validate that further.
Hey, so great talk. So a couple of questions. So one is just following up on what he was saying. Have you looked at what would be a minimal network that can give this behavior? Like, what is the smallest set of teams that you could have?
Two members. So 4-node network can give teams.
Okay. And no, but for a desired level of robustness, right? A comparable level of robustness or something. So how over?
So this is a two-team network. We didn't know this 10 years ago to call it teams, but exactly this is what we are seeing. But your question is right. So how do we define robustness with respect to perturbation to what? So those are important questions that we are still thinking about.
And you talked about doing those perturbations, and large fraction of the perturbations resulted in robustness and things like that. So you removed an edge, is it?
In some cases we removed an edge, in some cases we changed the value of the node from on to off or off to on.
Okay.
So those are structural and dynamical perturbations, both kinds we did.
So it's just a comment, phenomenal work, Mohit. I don't know how do you get all the time to manage such a big team and all the projects that you're doing. Wonderful work, and the kind of results that you showed seems very fundamental. The kind of questions that I've shown, that if it is generally applicable for any networks of any size, and that you initially suspected it to be potentially a coding flaw. To me also it would seem so, but if it is not so, then wonderful result.
Thank you very much.
Hello. My question is, you showed that there are two teams and the two teams can interact with each other. So did you look at all possible ways in which the two teams could interact together, or any specific way?
Yeah, that's a great point. So we also looked at scenarios when, let's say, you add impurity. So members within a team start to inhibit each other, and members across teams start to activate each other. So for that, there was a high penalty in terms of team strength. And going back to, I think, Ken Srinivasa's question, that why would networks have higher density if by 30% or 40% you can achieve the same thing? So absence of team members do not have as high a penalty, but impurity or infidelity to your team clearly has a high penalty in terms of functioning of the system. So those are the two things we have been able to dissect so far.
Okay, thank you.
So one of the things which I find striking about this EMT network is that you have this E and M states which are completely opposite of each other.
Right.
So you are having two attractors which have the maximum Hamming distance between them.
Right.
Is that important for imposing and getting the team structure?
In some extent, yes, because again, that going back to Rahul's question, because we ordered the nodes in such a way that team 1 nodes were together and team 2 nodes were together. So it need not be perhaps the strongest attractors, but clearly they need to be mutually antagonistic.
So if you impose attractors which are very close to each other, the team structure may not appear.
Right.
The other thing is that, which I have discussed sometime, is that how important is this democratic logical rule that you are imposing? Because what we observe is it's very different from that.
Right.
Because there is no veto of any edge on your network.
Right. So we have done some very, very preliminary work to address that kind of question, where all we have done is that we add weight factors to each of the edge. So we are still not changing the rules, but we are changing the weight factors. And the question we were asking is that now if you say have 1,000 such combinations or 100 such combinations of different weight factors, then how much does the phenotypic distribution change? In at least networks with strong teams, we do not see a large difference. We still see that the most frequent epithelial and mesenchymal states are maintained, although their dominance may go down and the frequency of hybrid states may go up. So that is what we have seen in very, very preliminary work so far.
No, what's striking about your small cell lung cancer paper is that most nodes have very high degree.
Yes,
So
yes,
that's,
yes.
that's very striking.
Right, so that's why we were not very confident about that network, because that network was derived via statistical methods, and I don't think there's extensive literature to validate every edge. But for EMT networks, these were all bottom-up built, so there's no sort of circular argument being made in that case.
The full talk.
Hi Mohit, nice talk. I had a suggestion, which is about your question of for a fixed matrix and two teams, what is the minimum number of the density that you need? So maybe you can look at some results in percolation theory.
Okay.
And that also shows this sort of transition. I mean, that is related to connectivity.
Right.
But this could be sort of connectivity within the teams and across inhibitions across the team. So that's
Certainly.
just a suggestion.
Sure, thank you, thank you.